For the fabrication of any biosensor, our biorecognition element or sensing element needs to be connected to a transducer. Remember that the biorecognition element could be an antibody, it could be a receptor, it could be a nucleic acid, an aptamer, etc. That sensing element needs to be attached to the transducer to be able to transmit the signal. And that attachment could be in one of these ways. It could be through passive absorption, covalent attachment, cross-linking, entrapment, or encapsulation. And we're going to be talking about each of these methods in a little bit more detail. The first one is passive absorption. It basically involves contact of the biorecognition molecule with the substrate, which is basically the sensing, the transducing system, for a long period of time. And basically allowing them to interact and allowing them to form non-covalent bonds, basically hydrogen bonding, hydrophobic interactions, etc. The benefits is that we're not doing any chemical reaction. We're just allowing the two to be in contact with one another and be able to absorb reversibly. And so it's simple, is typically gentle on proteins and enzymes because we're not actually doing any chemical reaction. We're just putting them together under mild conditions. And it usually results in the formation of monolayers, which it might be beneficial. The problems with this is that we don't have any control over the orientation by which these biorecognition elements are absorbing onto the surface. And so we could have different scenarios. For example, here for an antibody, as we mentioned, the FAB regions are the regions that are responsible for providing that antibody the ability to selectively bind to a target molecule. And so in this end-on FAB app, that would be the desired configuration. But we could similarly have all of these other configurations that are not necessarily going to be able to interact and bind to the target. Now, adsorption can sometimes also result in denaturation, which is misfolding of the protein or antibody or enzyme, and that's what's shown here on the right-hand side. Sometimes for the adsorption to be able to occur, that protein needs to unfold to be able to make available certain sites. For example, if the surface is very hydrophobic, protein adsorption would typically lead to unfolding to expose hydrophobic regions of the protein that are typically inside of that protein. And so because of this, you might actually denature the protein and not enable it to be to have any affinity now toward the target of interest. Oops, I apologize. Another problem with passive absorption is that it is not stable. Basically, that absorption is sensitive to desorption either over time or in response to environmental conditions such as changes in temperature, changes in the pH, changes in the ionic strength of the medium surrounding, or even competition from other molecules that might have more affinity toward that subsurface. Now, passive absorption can be utilized and it can be improved through a number of methods. One of the methods that we're going to discuss later is cross-linking of previously absorbed biomolecules. But another one is that we can utilize passive absorption in conjunction with affinity processes. For example, here we can pre-absorb a protein such as avidin or streptavidin, and then utilizing an affinity interaction between this protein and another molecule such as biotin, then we can improve the orientation of the biorecognition molecule. So in this case, we're utilizing the high affinity of biotin to either avidin or streptavidin to enable an attachment of an antibody. And by doing so, we are basically ensuring that the antibody, that the important part of the antibody is being presented toward the surface. Now, a note that I have here is that despite all of the limitations, passive absorption is still really important. It is actually typically the method of choice for any kind of preliminary investigation or for the fabrication of disposable sensors, just because of the fact that the drawback of poor stability might not be an issue, but the cost benefit might be sufficient to make it feasible or make it even the method of choice for some of these systems. The second method to immobilize biorecognition elements onto a transducing surface is the use of covalent immobilization, where we're actually doing a chemical reaction that will lead to a covalent bond being formed in between that biorecognition molecule and the surface of the transducing system. To be able to do this, we utilize bioconjugation methods, which can be utilized not only for immobilization, but could also be utilized in order to attach labeling molecules to the biorecognition molecule, or in order to cross-link proteins and enzymes to be able to protect them after adsorption. The benefits of covalent immobilization include excellent stability because you're forming a strong bond. The drawbacks is that it can be time consuming, it can require costly reagents, which might make it impossible for this to be a commercial product. And it can also affect the biological activity of that biorecognition molecule because sometimes the chemistry that is utilized could use some of the functional groups that are in the sites that are important for recognition of the target analyte. At the same time, some of these chemical reactions might require the use of unsuitable conditions, for example, high temperatures or the use of organic solvents, which might denature or affect the activity of that biomolecule. So because of this, we need to be very careful with what chemistries we utilize and optimize those chemistries to make sure that the covalently attached proteins are active enough after that immobilization. One thing that I want to mention before I move from this slide is that there is a great book called Bioconjugate Techniques that talks about all of the chemistries that can be utilized for covalent attachment of biomolecules to other materials. And it also talks about all of the applications of these immobilized molecules, including biosensors. This is a really great book. It is not for chemists necessarily. It's a great resource for anybody in this biomedical field. All right, so what functional groups can we utilize in order to enable immobilization of biorecognition elements? Well, there's actually not that many due to the specific molecules that are present in biological molecules. So to begin with, we have primary amines. Primary amines are present in every protein or polypeptide on the N terminus and also on all of the side chains of the amino acid lysine. Carboxylic acids are going to be present in the C terminus of every single protein and every polypeptide chain, as well as on the side group of aspartic acid and glutamic acid, which are two amino acids that are part of proteins and polypeptides. The next functional group is sulfhydryls, which are also called thiol groups. These thiol groups are found on the side chain of cysteine, which is an amino acid. These thiol groups can enable actually the formation of disulfide bonds in between different cysteine groups that might be on the same polypeptide or on the same protein. And this is actually one of the bonds that enables the formation of the secondary and tertiary structure of proteins. So it's a really important bond. But anyways, these functional groups can be utilized for us to attach those molecules to a surface. Hydroxyl groups are also found on the side groups of serine, threonine, and tyrosine, which are three different amino acids. They are also found obviously on polysaccharides and glycoproteins. And then finally, aldehydes, which are not actually normally present on biomolecules, but that they can be easily formed by oxidation of hydroxyl groups into the formation of these aldehydes. So now, knowing what functional groups are available on the biological molecules of interest, these are going to be the chemistries that can be utilized in order to attach those to the surface of the transducer. For example, for a carboxylic acid, we're going to be utilizing carbodiamide chemistry. For an amine group, one of the chemistries that can be utilized is N-hydroxysuccinimide esters. For sulfhydryls, malamides are going to be your molecule of choice or linker of choice, etc. I'm going to be going over a couple of examples, and I'm just providing here a list for you guys to have available in case that you need to do any kind of chemistry like this. Here at the bottom, I included bioorthogonal chemistries, which we had talked about when we talked about the methods to prepare hydrogels. These bioorthogonal chemistries are basically not utilizing any of the functional groups that are normally present in biological molecules. Instead, what would happen is that the biological molecule would be functionalized to or would be modified to be able to include either an alkyne or an acide, for example, to enable click chemistry with the other one. So for example, if we functionalize the biological molecule with an alkyne, that alkyne can then react with an acide containing molecule to form a triazole bond. Or reversibly, we could functionalize the biomolecule with an acide group and then be able to do a reaction with an alkyne through click chemistry as well. Now, in order to immobilize the biorecognition element, we're going to be utilizing linkers. In this first example, I'm showing a zero-length linker, which is going to enable binding of a biological molecule directly through just a functional group to the surface. In this particular case, I'm showing the reaction between molecule A and molecule B, where A or B could be either the biorecognition element, meaning the antibody, aptamer, or whatever, or the surface itself. So one of them would have to have a carboxylic acid, the other one would have to have an amine group. To enable the reaction, we're going to utilize carbodiimide molecule. That carbodiimide molecule is typically going to be either EDC, which is water soluble, or dicyclohexyl carbodiimide, which is not water soluble. These two react to form an active intermediate that is then reactive with that primary amine group, enabling the formation of an amide linkage and having a byproduct from the reaction, which is a urea byproduct. This is a very typical chemical reaction that is done in order to modify or functionalize or immobilize proteins onto surfaces, because remember that proteins have both amine groups as well as carboxylic acid groups. Another very similar strategy utilizes not just or let me back up, it's another strategy to be able to also form an amide bond in between two molecules, molecule 1 and molecule 2, where 1 and 2 could be either the biorecognition molecule or the surface. We're still going to be utilizing a carbodiimide molecule, such as EDC in this particular case, forming that reactive intermediate. But now, instead of directly attaching the second molecule with the primary amine, what we're going to do is that we're going to react this intermediate, which is unstable, with N-hydroxysuccinimide or sulfo-N-hydroxysuccinimide in order to make a new, more stable intermediate, which is amine reactive as well. So this would be called the sulfo-NHS ester if we utilize sulfo-NHS, or it would be called the NHS ester if we were utilizing NHS by itself. This intermediate can then react with the primary amine, leading to the formation of the stable amide bond in between 1 and 2. Now, the difference between these two is that the sulfo-NHS has this sulfonate group, which makes it much more water soluble. And so the intermediate that gets formed here is more water soluble and better to be able to attach to a biological molecule, for example, that is dissolved in water and is hydrophilic. On the other hand, this is also a much more expensive molecule, so sometimes cost makes us not be able to utilize it. In addition to zero-length crosslinkers, we also have bifunctional linkers that are going to provide a slightly or significantly larger spacing in between the substrate or surface and the biological molecule that we're trying to immobilize. Among these, we're going to have both homobifunctional as well as heterobifunctional molecules and some polyethylene glycol derivatives. All of these are going to provide the benefit of having significantly enhanced mobility for the biological molecule, which is going to hopefully also help with the ability of that biological molecule to be able to interact with the target analyte or ligand without so much steric hindrance. Among the homobifunctional molecules, I'm showing three different examples. In the first example, we have the binding of a molecule A to a molecule B, where again, A or B could be either the biorecognition element or the surface. And in this particular case, both of them are functionalized with primary amines, which can then be linked through this glutyraldehyde molecule that has two aldehydes that are going to be reactive toward amine groups, forming here a linkage, an imine bond through a little spacer. That spacer is what provides, enhanced spacing for the biological molecules and enhanced flexibility for the biological molecule. Another example here is shown through the reaction of an aldehyde-derived biomolecule with this linkage, which is a dihydroxide molecule. It's an adipic acid dihydroxide. Imagine that we have two molecules that have aldehyde groups on either end, and basically the reaction would occur on both ends. One of them would be the surface, one of them would be the biorecognition element, and what you end up forming is this hydrazone linkage. This linkage is actually interesting because it is a pH-sensitive linkage that is cleaved at low pH of about pH 5. So it's something that would enable you to have a degradable linkage or a pH-responsive linkage. The third example that I show here is utilizing this carbonyl diimidazole. And basically, this molecule is going to enable us to conjugate a hydroxyl functionalized molecule together with another hydroxyl molecule, or even with a molecule that has a primary amine, because notice that both sides are identical and they are reactive to both hydroxyls as well as an amine groups. And so in this particular case, as is shown, what we're doing is a two-step reaction where we first do the reaction between the hydroxyl and the imidazole group here, with the byproduct being imidazole. And then we do a second step where we react the second molecule to this other side, releasing another imidazole. The bonds that are formed are carbonate, or the bond that is formed in this particular case is a carbamate linkage that basically looks like you have an ester on one side and an amide on the other side. That again is known as a carbamate linkage. Notice that in these particular cases, all of these examples, the linkage is just a little bit longer than a zero-length linker. It provides some flexibility, but they are fairly short. In 2 slides, I will be showing some polyethylene glycol-enabled linkers that are going to provide a lot more flexibility. Here I'm showing some of the heterofunctional linkages that can be utilized. This is actually just one particular molecule called SMCC that has both an N-hydroxysuccinamide group that is reactive against primary amines, and it has a malimide group that is reactive toward diol groups. So basically this could be again utilized to be able to link a biological molecule by using its primary amines or utilizing its diol groups to a surface that would have the complementary functional group. In this particular case, we have molecule A and molecule B, where either one of those two could be the biological recognition element or the surface. And what they do is first the reaction with the primary amine, leading to the formation of the amide bond, which is a stable bond, and liberating N-hydroxysuccinamide. And then a second step, which reacts the diol group with the malimide bond in order to form a thioether bond right here. I should also mention that there's another variation of this SMCC molecule, which has a sulfur group on the N-hydroxysuccinamide, as we had discussed earlier, and that just provides it with more water solubility, but it's also a more expensive molecule, so it can be of limited use. Now, here are the polyethylene glycol or PEG linkers. So they're going to have similar functional groups on either end, either heterofunctional as shown here, or homofunctional, meaning that we have the same functional group on both ends, but the linkage is polyethylene glycol. In this particular case, it's just 4 repeat units, but it could be longer. And the functional molecules that we have on the ends could be malimide groups, as shown here, or N-hydroxysuccinamide groups, as shown here, diol groups, carboxylic acids, primary amines, And the last one that I think I have here is actually this biotin molecule that is also shown here and here, which could enable the attachment of that polyethylene glycol to avidin or streptavidin on either end. As you can see, basically all of these are going to provide significantly more flexibility than the short linkers that I showed in the 2 slides before this. And so these can be significantly more helpful if you're having steric hindrance problems, or if you just need your biorecognition molecule to be flexible to be able to better interact with the analyte of interest. One note that I put down here is that I listed some of the companies that are the major players in the field of selling functionalized polyethylene glycols. Lysan Bio is one of the important ones that is a US company, and it has a long history. It used to be called Nectar, and it's basically one of the first, it was one of the first companies selling functional polyethylene glycols. They are in Alabama, I believe. And then there are some other companies like Creative Pegworks, which are, I believe, is from China. And then Thermo Fisher has been more recently starting to sell some of those functional polyethylene glycols as well. All right, another method that we can utilize is cross-linking of the biorecognition molecule. So you could think about this as a hybrid between adsorption and covalent immobilization. What this means is that we're going to be depositing the biorecognition element onto the surface and allowing it to adsorb through non-covalent interactions. But after it has adsorbed, we're then going to cross-link it chemically in order to immobilize it in a more stable form. Because remember that we said that an adsorbed protein or an adsorbed biorecognition molecule could come off either with time or through interaction with competing molecules, or just because the bond is not very strong. So in this case, what we're doing is, again, we're first depositing it and then we are cross-linking it as is shown here. The benefits of this is that it can increase the stability because it basically prevents those molecules from just floating away compared to absorption. It can also increase the sensor response by enabling a multi-layer assembly, and that's what I'm showing here in this particular research paper. So they actually compared having a single monolayer of the biorecognition molecule that was absorbed versus a cross-linked system where now we can have a multi-layer that provides more of these biorecognition molecules. And they were able to show that the sensitivity of the cross-linked system was significantly higher because basically you have more ability to recognize molecules. And this particular sensor was one that is for glucose detection utilizing glucose oxidase. Now, the drawback of utilizing cross-linking is that you're basically also doing chemical reactions. And as we mentioned before, those chemical reactions can decrease the activity of the biological molecule because you might utilize some of the functional groups that are important for the ability of that molecule to actually interact and bind to the target analyte. So you always have to optimize your chemistry to make sure that that molecule, that biorecognition molecule is still active. The next method for immobilization of biorecognition elements is entrapment. The entrapment can be either in a matrix or in a porous network. Here I'm showing a matrix on the right hand side, which is basically a hydrogel, a polymer network, or a viscous solution. We have already talked about hydrogels extensively, but basically what you have is a polymer that is chemically or physically cross-linked, and because of the high porosity, we are able to entrap biological molecules like antibodies or enzymes within it. A separate one is going to be porous medium, which is talking about more solid kind of materials. And this porous medium could have pores in different size ranges. So a microporous system, also known as a zeolite, is going to have very small pores of less than 2 nanometers. A mesoporous system is going to have pores of about 2 to 50 nanometers, and then a macroporous system is going to have pores that are greater than 50 nanometers. The types of materials that form these mesoporous materials include silica, carbon, metal oxides, phosphates, ceramics, and semiconductors. And what I'm showing here is one of the ways to be able to generate those mesoporous structures. In this particular case, for the formation of a silica material. So what they do is to begin with a self-assembly of organic molecules into the form of either spherical micelles or rod micelles or a rod micelle assembly. And then they're going to surround this with the precursor for your silica, which in this case is tetratoxysilane, sorry. And the tetratoxysilane can then be reacted into a salt, it's a salt gel reaction. So taking a salt and reacting it to form a gel system. And that basically generates silica. And the silica is going to be surrounding all of the rod micelles in this particular example. And after we have this, we're going to be removing that that self-assembled organic molecule, which is basically a surfactant molecule to be able to form micelles. And that can be removed in either two ways. One of the ways would be extraction, and one of the ways is calcination, where we basically burn away all of the organics, leaving here the mesoporous material. On the right-hand side, I'm just showing an example of a mesoporous system in which they generate pores that are about 20 to 50 nanometers in size that enable the entrapment of glucose oxidase for glucose sensing. But at the same time, this mesoporous system has micropores that enables the quick and selective diffusion of the small glucose molecules through. And so with this system, they were able to show good sensitivity and good selectivity for glucose. The last of the methods that we use for immobilization of biorecognition elements is going to be encapsulation, which is basically the immobilization through enclosing that biological molecule within a vesicle or a polymer membrane. That membrane could be a selectively permeable membrane that basically only allows the analyte and the product to come in, but that prevents the biological molecule, meaning the antibody or the aptamer or the enzyme, from leaving, right? So only small molecules such as the analyte can come in, the larger molecule cannot come out. Now I put an asterisk here because in certain cases it might be very difficult to do that if there's not a big difference in between the size of the analyte and the biorecognition molecule, for example. And so in such cases, one strategy that you can use is to first kind of encapsulate the biological molecule into a very porous system, and then you can cross-link it in place, chemically cross-link it. Now that you have these cross-linked aggregates, they are much larger in size than the pores, and they won't be able to come out. Now, the encapsulation can be a good thing in order to stabilize the enzymes or antibodies or even whole cells. It can be these encapsulation systems can be either microscale or nanoscale, such as a liposome. And the encapsulation can be achieved through either an interfacial polymerization, in which basically you take aqueous droplets of your enzyme and you emulsify them into a monomer solution in an organic solvent. And then you carry out the polymerization so that basically the monomer is going to polymerize on the surface of the aqueous droplets. And you end up with a vesicle that includes an aqueous core where the enzyme is located. You could also use other methods such as layer by layer assembly or soldier reactions or hydrogel formation around those spheres, basically. Now, I wanted to switch gears and talk about some of the metrics that are utilized in describing sensor performance. This is a long list, but I think that is important. One of the things that you'll see being mentioned on sensor articles is the limit of detection. Limit of detection is the lowest concentration that can be reliably measured. And on the right-hand side, we have an equation that shows the y, which is the response of the level of detection, is going to be equal to the response of the blank. Blank is basically if you're just detecting buffer, basically, whatever the background buffer is, plus K times SB, where K represents a numeric value that is chosen according to the confidence level desired. Typically, is going to be 3. And SB is the standard deviation of the blank. So what we're saying is that the limit of detection is going to be equal to the sensor response upon just interaction with a sample that does not contain the analyte, plus 3 times the standard deviation. And that's shown here graphically. If this is the response of our blank, then our our limit of detection should be basically 3 standard deviations higher than that. The next thing is going to be the limit of quantification, which is basically at what point the signal differs from the blank by more than 10 standard deviations. And again, that's shown graphically here. So we want to say that this is the kind of the smallest quantity that we can reliably and accurately provide a quantity for. So from there in higher concentrations. That means that we are higher than the standard deviation 10 times. The sensitivity, it is very commonly erroneously thought of as the smallest quantity that can be detected, but that's actually the limit of detection. So what sensitivity actually means is that is the change in response produced by a unit variation in concentration. For example, if I had a pH meter, then, and let's say that it was an electrochemical pH meter, then it would be like how many volts does my intensity change for every pH unit. So it's a ratio of the response to whatever variation had happened, in this case pH units. The response range is going to be the range in which the sensor can accurately measure different concentrations. The lower limit is going to be the limit of detection. The upper limit is going to be the concentration at which the response deviates from a calibration function. For example, if you see that your system has a linear response, then the upper limit would be the point at which the data no longer follows that linear response. You start basically seeing a kind of a plateau of the data where it's no longer changing at the same rate. The ratio between the upper and the lower limit of detections represents the dynamic range for that sensor. Now, the resolution is the smallest detectable change in analyte concentration, and it depends on the noise level and the sensitivity. So the higher the resolution will be if you have low noise and high sensitivity. The resolution, you can think about it like basically what is, if I have a pH meter, what is the smallest unit of pH that I can see a difference on? Is it 0.001 or is it 0.1 or is it just 1 pH unit in between, in between two measurements? The specificity is going to be basically the ability of the sensor to discriminate a particular analyte from a complex mixture without interference from the other sample components. It is a combination of high selective separation and low matrix interference. When we talk about selective separation, we mean the selectivity of the biorecognition element for whatever analyte you are interested in. And then low matrix interference, we're talking about basically the blank. How does the blank behave? Does the blank show a high response for that system? The blank is talking about the buffer, whatever other constituents you might have in the system. Now, selectivity can be crucial in terms of being able to, in terms of your biorecognition element, being able to just detect one molecule. But that might be really difficult to achieve. So something that is being done nowadays is to utilize poorly selective sensor arrays, but by utilizing an array, you basically can obtain more information And utilizing data processing methods, you can actually collect very accurate information about what is present in the sample. For example, I might be interested in detecting just one biological molecule, one protein, and I can detect it by utilizing various antibodies that have affinity toward that one biological molecule. And so by using various antibodies, I can kind of overcome the deficiencies of one or of each of them and be able to get good, accurate data with poorly selective antibodies, if you would. The last thing here is response time, which is the time from the time that it takes for the sensor to be able to read after analyte is administered. Of course, when it says time to read, it means at what point does the signal actually become stable. So the time basically from administration to stability. One of the last things that I wanted to talk about was applications of biosensors. These are pretty straightforward, pretty common sense, but basically we can use biosensors for environmental monitoring, in which case we're basically going to be looking at pollution in air and water samples. Also in the food and agriculture and biotechnology industries, we might be looking at nutrient content, freshness of food, presence of pathogens, and also the presence of certain ions and molecules that might be in them. In the case of healthcare, we're talking about biosensors for in vivo or in vitro use. Of course, glucose testing is one of the most important ones, but also the detection of biomarkers of disease, including cancer, cardiovascular disease, or the presence of pathogens like bacteria or viruses, and other biomarkers of either normal or pathogenic processes in the body. And then in terms of defense, the detection of explosives, warfare agents, pathogens, or toxins is also important. In terms of the trends that we have seen over the last 4 decades, basically at the beginning, we were looking at the development of biosensors and focusing on the performance of those biosensors. So trying to improve the specificity, trying to improve the robustness of those systems, the cost, the simplicity, and the ease of use. Next, we're looking at integration. So how can we increase the functionality and the ease of use of these systems as well? And to do that, we're looking at utilizing maybe multi-step or reducing the number of steps in some cases, multiplexing, where we're looking at detecting more than one analyte. In this, an example of this would be DNA microarrays. We're looking to reduce instrumentation costs, and we're looking to reduce the number of, the credentials of the users that are required, right? Now, how easy can this system be run? Can it be run by just anybody? As of late, we're also looking at miniaturization to be able to utilize the systems in vivo, for example. And so we're talking about integration of biosensors into lab-on-a-chip systems or microelectrical mechanical systems. We're looking at utilizing or at developing point-of-care systems that would not require any kind of expertise to be able to run as well. So basically trying to make them more accessible. The last things that I wanted to show you, this is kind of for you guys to read, but it's basically the history of biosensors, showing you some of the milestones in the field. One name that I wanted to point out is Dr. Clark, who actually invented the first oxygen electrode and also the first amperometric sensor for glucose. And with that, I will let you look at this. And here's a picture of Dr. Clark. All right, have a great day, guys. Bye.